Guy Sella from Columbia University, who is going to tell us about the population genetic model for disease susceptibility and other cognitive traits.
So hello, and it's a pleasure to be here, and I'd like to thank the organizers for the kind invitation. So I'll begin with, for the benefit of the computer science side of the room, I'll begin with a brief introduction, and I apologize in advance for those who know this stuff inside out and for some vulgarizations in order to get everybody up to speed. And another apologetic initial comment is that I'll focus on examples from humans, which is not in any way to underestimate 100 years of work on other systems. Shortly after the publication of The Origin of Species, Darwin's cousin, actually Francis Galton, became obsessed with studying human genetic variation, and he soon discovered that many phenotypes appear to be normally distributed high in natural population, well, in human populations. In this case, you see the histogram for height. Another observation he made is about the relationship between parents and offspring, or more generally relatives. And this is a famous plot from Galton where you see the height of the offspring as a function of the average height of the parent normalized for males and females, where the size of the points actually denote the sample size, and this line is just the y equal x, which is what we would expect to see if the correlation was 1 between the two, and this is the regression line, which is lower than 1, which led Galton and others to develop many important concepts and coin some terms you all know, like nature versus nurture, or regression to the mean. And it took quite a while, and Ronald Fisher, to come and explain how these observations could be, and others could be explained in terms of Mendelian inheritance. In this seminal 1918 paper on the correlation between relatives and the supposition of Mendelian inheritance, Fisher showed that these observations and others could be explained in terms of a simple additive model, where I wrote a version of this model here, where we assume that the phenotype has a genetic and environmental contribution, and more specifically that the genetic contribution, we could break it up into the contribution across loci, and where here you have the contribution of the allele from the maternal and paternal sides, and this is the environmental contribution. And if you assume that the environmental contribution is normally distributed, and that the contributions, the additive contributions across loci, that there's many of them and they're IID, then you get the normal distribution of phenotypes in the population, where it's the sum of the additive genetic contribution and the environmental contribution. Just to give you an example from humans, so these are a bunch of SNPs that were found to be associated with height, the top 20 SNPs for height, with their effect sizes in males and females normalized in some way. The typical effect here is 2 millimeters, just to give you a sense. And here what you see is the number of tall alleles, which ranges between 0 and 40, because we have 20 loci in the 20 biallelic loci in this case. And here is the histogram, the percent of the sample with different number of alleles. And here is the relationship between the number of tall alleles and the height difference, again with normalization, and it appears to be approximately linear. This is, of course, not a rigorous test of the additive model, but at least it corresponds with its predictions. A second thing you could do with this additive model is you could define heritability as the fraction of variance that is explained by the additive genetic contribution. And once you do that, Fisher showed that in this specific case, you could show that the slope in this figure is simply this heritability, and more generally derive the relationship between pairs of relatives of different relationships. Now, you could measure heritabilities until recently, according to correlation between relatives, and twin studies and so on. Now there's perhaps better ways to do that. And the surprising thing is that heritabilities tend to be extremely high for a wide variety of traits, typically between 10 and 90%, where here you see some examples for humans and specifically European populations, and here you see some examples from other systems. There's also some patterns that emerge that I won't go into. You see here the morphological and fitness traits show different patterns. We don't have a good explanation for this, neither do we have a good explanation for why heritabilities are typically so high, or why there's so much quantitative genetic variation. Now, how do we go about finding the variance? A moment ago I showed you some variance for height, so one way you could go about it, which I'll focus on today, is a genome-wide association study, which, owing to massive efforts over the last decade, led to the discovery of around 10,000 variants corresponding to hundreds of traits, including the susceptibility to various complex diseases. And the basic idea, at least, is very simple, where you take a large sample of individuals, you phenotype them, for example, you measure their height, and then you genotype them at many variable sites, and at each site you look at the 3 genotypes and at that relationship to the trait value, and then you do a regression, and the slope gives you the effect size on the trait, and then you get a p-value, and you need to correct for you need to adjust for false discovery rate. So you use these p-values. This is how it's usually represented in terms of a Manhattan plot, where this is the log p-value, and this is just the chromosomes of the human genome, with each point here corresponding to a p-value for a given SNP, and this is the threshold for genome-wide significance in this case, and you see a significant association. So just to continue with the examples of height, so the giant consortium did this, of course, with some statistical sophistication, for more than 180,000 individuals, genotyping more than 2 million SNPs for each individual, and discovering 180 associations, loci associated with height, where here these associations are represented in terms of the minor allele frequency for this SNP, and here is the effect size, and this line corresponds to some level of power to discover this in the association study. So you get these kind of scatter plots in allele frequency and effect size.
So this
And
is for
yeah.
for a single SNP, all this plot for a
The,
single
no, this plot is all these points are all the genome-wide significant SNPs to come out of this association study. So each point is a single SNP that turned out to be a significant association.
So presumably there's SNPs under the curve that are really there.
We'll get to that. Give me 1 minute. So, okay, so yeah, so obviously there's limited power, and indeed when you look at, you could estimate how much of the heritable variance in height do these associations explain, and the hatch is despite discovering 180 associations, this explains only 10% of the heritable variation, and this is, the well-known from recent years missing heritability problem. So now we get to define genetic architecture, at least the way I'm going to use it in this talk, and the genetic architecture is usually defined in this field in terms of the number of all the variants in the populations, not only those that we discovered in the association study, and the joint distribution of their frequency and effect size. Okay, so that's a definition I would use. Of course, in a given as Lee pointed out, in a given association studies, you have power limitations, but you could still begin to compare architectures among traits. So you could look at the discovered number, you could compare the distributions. You need to be mindful of the power and the study design, and you could compare how much of the heritability was explained for different traits. So you could begin to look at differences between the architecture of different traits. And here are some examples, where here is the example of height that we've seen before. Below is an example of a body mass index. And here is an example of schizophrenia, where I should just note that given in this case we're talking about the probability of developing the disease, then the effect size here actually corresponds to the log odds ratio that was found in the study, which is the equivalent of the additive effect size for the simplest model for disease susceptibility. And when you look at these plots, you could already begin to see some differences. So for example, if you compare height and BMI, and I note here that BMI is actually a larger study, at least in terms of the sample size, then you see that much less heritability was explained in BMI as compared to height, and there's a much fewer variance. And you could also look at the joint distribution and begin to compare it, and you see that, at least for schizophrenia, that this joint distribution appears to be to follow kind of tightly around the power line, whereas for height it still follows it, but there's a bit more of a spread. And for BMI, it follows this even less so, where specifically we have these variants which are at high frequency and large effect size. So this leads to a number of questions. So one question is, we would like to know how genetic and population genetic processes shape the genetic architecture of variation in quantitative traits. For example, we would like to know whether we expect to see a different architecture and how for early onset severe diseases or for anthropomorphic traits. Another thing which is of practical import is, if we have some idea of how that behaves, perhaps we'll be able to predict or inform these study designs. For example, how much would you expect to gain in terms of explained heritability and number of variants if you increase the study of your genome association study twofold? It also relates to very basic questions that came up a few times during this meeting, which has to do with the fact that, you know, despite most phenotypes we care about being quantitative, we still know little about the processes that shape them. So maybe we could rely on these genome-wide association studies to do some inference and learn something about these processes and underlying parameters. And last but not least in this list, if we think about if a lot of phenotypes that evolution cares about are quantitative and there's a lot of variation, you would expect that when the environment changes, then the selection response would come primarily from this from the genetic variation. In order to understand these processes that have been recently called polygenic adaptation, then I think you need to know something about the architecture. So I'm actually going to talk only mainly on the first question, and I'm going to touch upon the second and third, and this is also the point to say that this is work very much in progress. So just to give some basic intuition to begin to model these questions. So if we think about the joint distribution of frequencies and effect sizes, so by and large we can think of the frequency of alleles to be determined by selection and drift. So here you see the frequency spectrum for different scaled selection coefficients. And now for architecture we want to know how these frequencies relate to effect sizes on our trait of interest. So we could think generally about two extremes. One is a case where the selection coefficient is independent of the effect size. So how could this happen? This means that our trait does not have a substantial effect on fitness. However, individual variants could affect other phenotypes and therefore have selection on them. In this case, if we look at different effect sizes, then we expect to see the same frequency spectrum. The other extreme example is if we have a phenotype that actually is the main source of selection on the variants that contribute to it. And in that case, the effect size will determine the selection coefficient, and you expect to see a strong relationship between the effect size on the trait and the frequency in which you find the allele. So just to give you, some examples, so perhaps one could argue that if you think about early onset diseases that are severe or diseases that affect fertility, then maybe they're closer to this extreme of determining the selection on the variants. And if you think of anthropomorphic traits, maybe height or BMI, maybe they're closer to the pleiotropic extreme that I just mentioned here. So this gives us an idea that the selection coefficients on variants and the degree of pleiotropy are important determinants of genetic architecture. So now we could go ahead and try to model this, and I'm just wrote down some requirements we would like to have in this model. So we would have to be able to have some parameter for the degree of pleiotropy. It would be nice if the pleiotropy will actually come from a description of some multidimensional phenotype, so we understand where it comes from. It would also be nice if the selection will come from this multidimensional phenotype as well. And of course, it's kind of nice if you actually know how to solve for things you care about in this model. So lucky enough, Fisher developed a model that fits the bill for some of these things, even though it was for different purposes. And this is Fisher's geometric model, where you consider where the trait space is an n-dimensional Euclidean space, where each dimension corresponds to a different trait or a different phenotype. So our individual is just a point or a vector in this n-dimensional space, where when we're going to look at this model, we're going to focus on one dimension, on the projection on one phenotype, but the dimension of this space will be a parameter for pleiotropy. Okay? Now, we could define fitness in this model. So fitness in this model is defined to be the optimum is at the origin, and the fitness declines as you move away from the optimum. Specifically, we're going to use, for analytical reasons, this Gaussian form, where here this W actually quantifies the strength of selection when W is smaller than selection is stronger. And I'll just note that even though this is a specific form, as long as the population is tight around this optimum, which will end up being the case, then this model would be equivalent to any model that has a quadratic form around this optimum. And now we'll introduce some population genetics into it. So first we'll introduce mutations. So we're going to assume an infinite sites model, which mainly means that mutations are introduced at some Poisson rate per individual per generation, and mutations never occur at the same site, so we're neglecting multiple mutations at the same site. Now, for mutations, we're going to assume that the mutation is the effect of the mutation on the phenotypes is a vector in this n-dimensional space that just describes the effect on its projection describes its effect on the different phenotypes. And we're going to assume that the size of these mutations come from some distribution, and their direction is just isotropic in this space, so chosen randomly in all directions. Lastly, we're going to do the standard Fisher sampling with viability selection, mutation, free recombination, and so on. So it's essentially the standard model. And just let me tell you how the genotype is related to the phenotype in this model. So if we have in individuals a phenotype, I didn't note, by the way, I should note as an aside, I forgot to note that the reason we don't have the environmental contribution is that it could actually be swallowed into the selection parameter. So in any way, you have an individual phenotype, and you could break it up into the contribution of fixed sites and the contribution of segregating sites. Now, for the segregating sites, so if we look at the contribution to the phenotype from segregating sites, where the I denote the individual, then because of the infinite sites model, each segregating site is biallelic. So what we do is we just sum over the additive contributions, where this GIL is just the genotype at locus L, which could take values 0, 1, or 2, and you could think of A as the derived mutation at this site. So it's a simple additive model. What we're going to do for the fixed contribution is that in our model, occasionally a mutation is going to fix, and we're just going to do bookkeeping. So we're going to update the fitness component, the phenotype that comes from fixed sites. Okay, so this is the basic model, and
So I guess I'm a little confused. So in this, the dimension of the phenotype space has somehow gone away.
No, because these are vectors.
Okay, each of those is a vector in this
N-dimensional space, yeah.
N-dimensional space.
Yeah, it's still additive. It's just additive in the n-dimensional Euclidean space. So Okay, this model fits some of our requirements. We have a variable degree of pleiotropy, which is the dimension. We have pleiotropy arises from the multiple phenotypes, from the different dimensions that we have, and selection results from how the selection acts on the phenotype, namely wanting to be at the origin in this n-dimensional space. So now it would be nice if we could actually calculate something, and despite the fact that this is quite a Baroque model, it turns out it behaves rather simply. So under sensible conditions about the mutation rate, namely that it's high enough such that it would be a quantitative trait, that many loci would segregate, but it won't be too high such that you would have multiple mutations affecting the trait arise in a single individual in a single generation. So under these conditions, it turns out that if we look at the distribution of phenotypes in the population at steady state, then that's described by a tight normal distribution around the optimum. Okay? So we're specifically the variance of that distribution is much smaller than the selection coefficient. And so why is this the case? Just to give you a simple intuition. So the fact that this is normal shouldn't be surprising because we have an additive model for the genetic variance. So if we assume that the fixed component is the same for the whole population, and the segregating component is a contribution is a sum over IID contributions at many sites. So that gives you the normality. Perhaps more interesting is why is it tight around the optimum? And the reason for that is that this system turns out to be equivalent to the population mean is equivalent to a ball connected to a string, where the string is connected to the optimum, but you have these, it's in a thermal environment. You have these particles actually hitting it. So the spring is equivalent to selection. The more displacement you have, the stronger selection becomes. So you can't wander very far away. And these fluctuations correspond to mutations and drift. And it turns out that this is actually not a metaphor. This is actually a precise analogy. And you could actually calculate the variance in the population mean, and that turns out to be small as long as the dimension is much smaller than the population size. So you could assume that you're tightly around this optimum. Now let's consider an individual allele, because we're interested. We're interested at say in a segregating site. We're interested in a variance. So let's consider a mutation that has effect size A, which that's a vector, and a frequency Q in the population. And given that we know the distribution of phenotypes in the population, we could now look at the distribution of background. So I focus at this particular locus, and then I look at all the contributions in the background, what their distribution is. And I know that the background plus the contribution of this locus needs to be normally distributed around the origin. So that gives me the distribution of backgrounds. Once I have the distribution of backgrounds, what I could do is I could calculate the fitness of each of the 3 genotypes in this locus. What I do is just I take the fitness of the 3 genotypes, but for that I need to know the background, but I integrate over the contribution of the background. And once I do that, then I could calculate the first 2 moments of change in allele frequency in a single generation. So the first moment is shown here, and the second moment is I'll get to that in a moment, and the second moment is just a standard second moment under a genetic drift. And once you have the first 2 moments of change in allele frequency in a single generation, then you could use the diffusion equation in order to get at least the closed integral forms for anything you'd like to calculate in this model. So, okay, so we can solve for things. Now, before we start solving for things, let's focus a second on the first moment, because there's some interesting things about it. So if you look at the form of the second moment, you see that this is, if you just put the parameter S over 2 here, this is just the standard form for the viability selection model with underdominance. Okay, so what this means is that frequency half at the site is an unstable fixed point, and essentially the rare allele is always pushed towards extinction in this case. Now the reason isn't a heterozygote disadvantage like in the standard viability selection, because heterozygote disadvantage doesn't even mean anything in this model. The reason is because the population is tightly centered around the mean, then the allele frequency at a given locus is actually selected to reduce the variance that it contributes to the phenotype. So you're trying to reduce the variance that this allele is contributing to the phenotypic variance, which is just in this model half times the mutation size squared times PQ, and this variance is maximized at half and minimized towards the end. So selection to reduce the variance just pushes this towards the end. So this is why you get this under a dominant form. It also suggests that it's probably a more general property of many of these stabilizing selection models. This was, by the way, shown for the one-dimensional case by Robertson in '61. Another thing that's interesting here is that it actually relates a selection with the size of the mutation, because you see the role of the selection coefficient is just played by this a squared divided by 2w squared. So this is the this is the selection coefficient here. So the size of the mutation in the n-dimensional space is directly related to a selection coefficient, and specifically, it tells us we could use natural units of 2n times this, which is just the scaled selection coefficient. So we have kind of like intuitive units for what goes on here. So now let's go to the kind of things that we care about, or at least that motivated this model. So if you recall, in the beginning I told you that the relationship between selection coefficient and effect size at our focal trait at the one dimension is likely going to be crucial for determining the genetic architecture. So it turns out we could actually calculate what this correlation would be under this model, and this is the form you get, where n is the dimension of the space, and this is the coefficient of variance of the distribution of selection coefficients, or mutation sizes, as I said a moment ago. And so, at least with respect to pleiotropy, this fits our intuition. So when the dimension is 1, then this correlation becomes 1. Okay?
The definition of the selection coefficient in this model is the apparent selection coefficient?
Yeah, it's exactly, it's equivalent. This is just this a square over 2w square. Okay, so what I call selection coefficient is just this a square, where a here is the norm in the n-dimensional space, divided by 2w squared, which was the selection parameter in the model. Is that okay?
So this distribution is just coming from the distribution of the distances from the peak.
Yeah.
Okay.
Yeah. So it turns out to simplify quite nicely in this case, and so we see that when there's one dimension, no pleiotropy, this term becomes 0 and the correlation is 1. You see that when you increase the dimension, the correlation goes down, but perhaps a bit surprising, at least at first sight, is the fact that even when you take the dimension, the pleiotropy to infinity, the correlation doesn't necessarily go to 0. And what you have is a term that depends on the coefficient of variance of selection coefficients. Now, upon second thought, this is not that surprising, because if you have a peak distribution, like a delta function distribution of selection coefficients, then actually the size of the mutation or the selection coefficients tells you nothing about the projection on the trait you're looking at. Only the direction determines this projection. However, if you have, so in that case, the correlation will be 0. However, if you have a wide distribution of selection coefficients, then larger mutations will actually tend to have larger projections on the trait you care about. So therefore, you would still maintain some correlation between selection coefficient and effect size. And here you see an example of this correlation as a function of a number of traits and different coefficients of variation for the distribution of selection coefficients. So now we could go and look at what we looked before, namely what's the relation as a function of these two dimensions. So here I increase the dimension of the space, the pleiotropy. Here I increase the wideness of the distribution of selection coefficient, the coefficient of variation. And in a one-dimensional model, then the relationship between the effect size on the trait and the frequency is the same. Does not depend on the coefficient of variation in the one-dimensional case, because the size of the mutation is exactly the size of the effect in one dimension. However, when when you move to a larger dimension, when you increase pleiotropy, when you increase the dimension, then you see that there's less of a relationship. The relationship is weaker between the effect sizes and the frequency spectrum. And then when you move to narrower distributions of selection coefficient, the relationship becomes even weaker. So this is what we saw at the beginning. And then one thing you could do is you could simulate an association study in this model. So in this case, we simulated an association study with the same parameters, but we adjusted things such that for a study size of 10,000, the discovered variance in all these 4 cases would be the same. And then we also adjusted the target size, how many mutations affect the trait, such that we'll discover the same number of variants in this case. So this is the power that, this corresponds to power half in a study size of 10,000, and this corresponds to a study size of 5,000 and 40,000, just to give you a sense of what's going on here. But more informative than just looking at these scatter plots is actually to look at the density of variance that comes from different combinations of effect size and frequency. So here the different shades of blue correspond to the regions that account for 20% of the variance, 40%, and so on, up to 95%. And if we start for the peaked case with one dimension, then you see that there's obviously in this case, there's a narrow distribution of effect sizes. And to understand the distribution along the frequencies, I just note that if we were in neutrality, then the contribution to variance just ends up to be the same at all frequencies. Because we have weak selection here, then we actually have more density towards the lower frequencies. So this explains the shape of the density here. Now, then when we move to a higher dimension, increase the pleiotropy, what we get is this widening of the distribution of effect size on our focal trait. And importantly, we also get a lot of variants that have very low effect sizes and or low frequencies, which, by the way, would be very difficult to capture in a genome-wide association study. And all the more so when you move to the case where the distribution of selection coefficient is actually wider. So this begins to tell you on how these different parameters would affect our discovery in genome-wide association studies. So here what I have is the expected explained variance in percents as a function of study size for these choices of parameters. And what you see that happens is for the case where you have no pleiotropy, one dimension, and a very peak distribution of selection coefficients, what's going to happen is until a certain study size, you won't discover anything, in this case, around 3,000. But then once you discover the first variance, you're very quickly going to discover all the variants when you increase your study size. When you move to the other extreme, which is the wide distribution with a lot of pleiotropy, then you're going to discover things with much lower sample sizes, but you'll have to go on increasing your sample size a lot in order to get to the tail of these things that have low effect sizes and or low frequencies. So this tells us something about how these parameters would affect how successful we expect to be in genome-wide association studies. Now, of course, you would like to do this for the real traits. You would like to, one thing you would like to do is you would like to do a inference of these parameters based on what you see from genome-wide association studies, and then maybe you could say, with the caveats of the assumptions of the model and so on, maybe you could say something about what you expect to see if you increase your sample size and how that's expected to vary in different traits. Take what I'm about to say with a huge grain of salt, but I recently heard that for schizophrenia, the consortium is about to publish a huge study where now, by increasing the sample size, but they managed to discover hundreds of variants, and that kind of fits vaguely, though you would like to do this more rigorously, to where schizophrenia fits in our two-dimensional parameter space in terms of kind of following tightly the power relation, but of course you need to do this rigorously. Another thing you could do with these kind of inference apropos is inform, get some information about what evolutionary parameters are perhaps shaping the variation here. Now, we're working on it. I'm not ready to show any results on that. I could just show you the basic idea of how we're going about doing the inference. And the basic idea is just that you could write down the likelihood of the model parameters here, which is the target size for this trait, some parameters of the distribution of selection coefficients, and the dimension in this specific model. And then you could write down this likelihood as a function of the discovered variance, the missing heritability that you didn't discover. That's an important input here. And also, of course, the study design. How big was the study, for example? That's quite important here. And and what you get when you write down this likelihood, then you just get the product of the probability of having a variant with a particular effect size and frequency times the power to detect this variance, and then there's a term for not explaining a certain fraction of the variance. So,
sorry, what happened to the prior probabilities of the parameters in the model?
So what we're doing is just maximum likelihood, so it's equivalent to assuming a uniform prior, I guess.
What was K here?
What was K here? I don't remember. Well, K I guess is what's discovered in the study. Why we put subscript K here, I don't remember at the moment. That's just, this is the total genetic variance in the trait minus the variance that you discovered.
It's the
in
number
the
of
association.
associated variants.
What?
I think it's the number of variants you say are associated with.
Oh, yeah,
Yeah.
you're right. This is the K here. So it's just what's explained by the K associations you found. Okay. So a brief summary. So we solved the model that I hope is intuitive for how pleiotropy and the distribution of selection coefficients affect architecture. Now, of course, you'd like to ask about other models for architecture. So even you could imagine many other models in the case of even in the case of stabilizing selection, and I think some of the things about these solutions will remain the same. Some maybe remain qualitatively the same. So, for example, the underdominance is likely to be quite a result. Quality being tightly around the optimum is probably a strong quality. I think you could use this type of approach, not that it's so complicated to solve if you write down other models. And regarding the qualitative, the results that I showed you of how things vary with pleiotropy and the distribution of selection coefficients, so there we're still grappling with trying to understand to what extent how this would change if we change specific assumptions of the model. Needless to say, there's also other models for how you get a quantitative genetic variation in a trait. So one is a parent stabilizing selection, namely the trait that we're talking about has no effect at the fitness at all, but the variants themselves have pleiotropic effects on other traits which are selected for. So one needs to look at that. And then there's also examples of models of balancing selection that reflect some trade-offs, and there's definitely some human traits, for example, traits related to autoimmune diseases, for example, that seem to be perhaps some balance between, humans, you move to more dense areas, the pathogen load increases, you wanted to increase the activation of your immune system, but then you suffer from autoimmune diseases, for example. This is just a hand waving. So these are other types of models one would want to look at. And inference, as I told you, or hopefully I'll be able to say something soon enough about that. And polygenic adaptation, I just don't want to go over time. So if you ask me about it, I'll tell you why I think the question of architecture is related to understanding polygenic adaptation. So let me just finish by thanking the collaborators on this work. So Yuval Simon's a student, no relation, a student in my lab, who did really the heavy lifting of the simulations and a lot of the analytical work here. Kevin Beloy, formerly at the University of Chicago, who was involved in the beginning of this work and now moved to pursue other passions of his, and Dick Hudson, who just made us smarter than we are. So that's all. Thank you.
Can you say something about polygenic adaptation?
Thank you. So I'll just give a hand-waving argument. So just to put this in context, so say a small population of humans leaves Africa and goes to live in much colder places, okay? And now you want a different ratio of surface area to volume, according to the Alan Bergman rule or some other rule like that. And obviously, because there's a lot of natural variation in phenotypes like that, then it's likely, at least, that the fast response of selection would be out of this natural variation. But then, if you want to ask about the basis of adaptation to such a shift in selection pressures, then you want to know, for example, where is the response going to come from? Where's the action going to be? Is it going to be from weakly selected sites that because they have small effect sizes, they were segregating at high population frequencies, and then they were responsible for the shift when you moved? Or are there going to be the strongly selected things which are at lower frequencies but have a bigger effect on the trait? For example, that's one type of question. Now, obviously, as I just talked about, the relationship between the frequency they're going to be in before the shift in the environment and the effect size on the trait depends on pleiotropy. So in order to understand how that's going to work, so here's a figure from a paper by Divladar and Nick here, which kind of like gives you a qualitative picture of, I think, a good initial way of thinking about what would happen. So here what we have is we have some quantitative trait where it was in the optimal value before we change the environment at time 0, and then it shifts to a different optimum. And then what happens, here you see what happens to allele frequency. So at the beginning, those with the right direction go up very quickly in order to match the new population mean. So here you see what happens to the trait mean. It very quickly matches the new optimum. However, they rise to a certain frequency, they match the mean very quickly. Now, some of them continue all the way to fixation, but that's probably a minority. But in order to balance their effect, the other ones go down back to their steady state probability. And this is why we see this transient effect on the variance in the quantitative trait. So at the beginning, the variance goes up a lot, and then selection effectively reduces the variance. But if you want to know which of these variants, what effect sizes, what selection coefficient they're going to have, are the ones to go all the way up, statistically speaking, then I think you need to know something about architecture. And of course that would affect other things, like what type do we expect to see, what type of signatures of linked selection, of sweeps we expect to see when you have a shift like that. And maybe a lot of human populations are actually that moved around are actually now in this transient state over here, which is why we don't see a lot of high FST in SNPs in the human genome. So, but I think that to get about it, this is at least how we got to study this question to begin with, is we were trying to do simulations of this, and we realized that we need to know something about architecture in order to go about this question.
So when this sort of stochastic theory was developed, it was based on really the idea there's an arbitrary relation between selection and allelic effect or trait. And what you're saying is, well, if we take Fisher's geometric model of stabilizing selection seriously, then that predicts a particular relationship. So you could phrase the question a different way and say, what could we observe in data to show whether the joint distribution of effect sizes and selection coefficients or minor allele frequency, what would that tell us about whether stabilizing selection is a plausible model? I mean, I have no idea whether it makes sense to think of these fictional traits, we're making them up as a high-dimensional quadratic space.
I totally agree. I think, one needs to get started somewhere, and, as you know, probably better than anyone, there's been, maybe 80 years of work in evolutionary quantitative genetics trying to get at these questions. primarily get at the question of why there's so much quantitative genetic variation. But there's been very little work with this, with this twist of trying to understand, how it would affect architecture. So this is definitely a starting point, and you would like to know first within models of stabilizing selection how much things would be sensitive to changing the assumptions of the model. So you could use the kind of models that Keatley and Hill used, for example, of some Y-chart distribution and other kind of things. So and you just want to understand how the observations would be sensitive to it, how your qualitative results would remain, and we're in the process of doing that. And I agree this is one model. It's definitely a toy model. We don't think that phenotypes behave like an n-dimensional Cartesian space, and there's other assumptions that go in here. What we like about this model as a starting point is that you kind of like intuitively understand what pleiotropy means, what selection acting on, and so on.
So another comment about the inference side. You could argue that you're doing it in completely the wrong way by focusing on those in the outline tail that have been ascertained. So you could say, well, what you're predicting is the distribution of minor allele frequency and effect sizes, but it's the bulk of the distribution that matters. And there's kind of funny things going on that are likely to be especially affecting the tail, which is what we see. So you could say we want to make inferences based on the bulk data. So
I agree. I think when you do inference, this kind of population genetic inference in general, when you're trying to infer the distribution of selection coefficients on amino acids, for example, then you have the problem that if you assume some parametric form, and if the data you have is kind of like highlighting a specific part of your parameter regime, then what gives you the ability to infer what happens in other parameter regimes is just the supposition of your parametric form. And I agree that this is definitely an issue. We focus here on the things that we have power to discover, and therefore, for example, I would be hesitant. It kind of like depends. You would like to try different distributions, but that's a cheap cop out. But I guess I'd be more inclined, assuming that robustness works out and so on, to believe inferences that have to do with what happened if you're going to, double your sample size in genome-wide association studies, as compared to inferences that have to do with how much of the variance has to do with rare variance, for example. Exactly because you're kind of like, extrapolating based on a different region in the parameter space. So I totally agree with that's a serious issue to contend with.
A last question. There's a slide you had where you were predicting these curves as you increase sample size. Just one before, yeah. So there seems to be discrepancies. You've got a study size of 100,000 and you found all the variants, but we've only found 2% of the variants. So all your different models are predicting that 100,000... Yeah,
but this is just the, this is an illustration figure. I chose specific parameters to generate this, this 2x2 of studies. I could easily choose different parameters that, you wouldn't find a lot in 100,000. And
so what do you have to assume then to get something with the reason, fitting estimate?
So there's a few things you could do. One rather So in terms of the So I think, essentially if you vary these parameters such as the, the degree of pleiotropy, the width of the distribution of selection coefficients, and also where the mode of that distribution is, then you could pretty much get with a given sample size whatever you want. And in terms of number of variants, and you just change the target size. So that would change the fraction discovered, but would change the number of variants you would discover. It would change. Maybe it would change the fraction a little bit, but not a lot.
It's just puzzling because these curves look very robust. If you made n 10,000 extrapolation of your curves, you're still not getting near the real regime. So it's something else varying. It's not just
but I think if you make, for example, if you make this wider, then I think it's going to change things quite dramatically. And if you you know, at the moment I'm assuming a 2NS of 3. If you assume a 2NS of, you know, 4, I'm assuming you're already going to discover much less of the variance. So this is just an illustration, and not necessarily, as I said, it's work in progress, where you'd like to choose different parameters to illustrate this dependence. So it's very qualitative, what we showed you here.
More questions? Thanks.